Hey biology students, it's Professor Gideon again, and we are doing the Biotechnology Lab today. This is a fun one, and if you enjoy crime scene shows like CSI, Law and Order, Dexter, you have probably actually seen this on TV. So as shown on TV, we're going to do the real science today. So I always like to have my students actually start by reading the crime that we're going to solve. So would you please open your lab manuals to page 128. Let's take a look. So at the top of page 128, we learn about the crime scene. So we've got all of our tools for this experiment, and we're going to first actually pour a gel. So if you can see here, I've got what looks like sort of a plastic tray with these rubber stoppers on both sides. And we use this to pour an agarose gel, okay? And I'm going to set that down a minute here. Just adjust the camera. Okay, and so what we're going to do is we're going to pour the gel. And normally what we have you guys do in the lab is you microwave these little flasks. And it looks like it has some sort of white solid liquid or solid material in there. This is what we call agarose. And you do microwave this for about 30 seconds. And when you microwave it, it becomes clear. It becomes a clear liquid. So I've already done that step. And here is the microwaved agarose. As you can see, it looks like a clear liquid. And I always tell students this is a lot like pouring Jello into a mold. If you've ever made Jello at home, you know that you have to heat it and it forms this nice liquid. And then you pour it into some sort of casting tray and then let it solidify. So we're actually going to do that here. We're going to be pouring what we call pouring a gel. So I already set it up by having this little casting tray ready to go. We also add this little blue tool here. We actually call it the comb. You can tell in the camera, it looks like it has little teeth in it, like you might use, for combing your hair. But this is actually going to allow us to form wells or little indents in the agar cast that we're going to make. So we push that down into the tray, snap that into place. Okay, so now we're going to take this liquefied agarose material and we're going to pour it into the casting tray. And it is a hot liquid. And when I pour it into that casting tray, it is going to sit there for about 15 minutes and until that solidifies into a gel material that we can use in the experiment. So I'm just going to set that guy off to the side there, okay? And then we're going to process 3 samples for this experiment. And these are the samples that we're going to process, okay? So you can notice they are in very small little miniature test tubes that we call microcentrifuge tubes, but the name isn't really essential. The point is these are really, really tiny, and that's because the amount of material we're working with here, the DNA we're working with in the experiment, is microliter samples. So in the past, we have measured milliliter samples using, say, graduated cylinders and so forth. These are very tiny amounts of liquid that we're using. And to transfer this liquid around, we use a tool here that we call a micropipette. You can see that there in the camera. This is called a micropipette here. I don't know if anybody's ever used these before, but this is used for transferring microliter quantities of DNA, which is what we do in this experiment. Okay? So here we have 3 samples. So we have sample 1, sample 2, in sample 3. And of course, I should mention where these samples are coming from. Now, they set it up in that demo there, or in the explanation. So sample 1 is the suspect's DNA. Okay, this guy is very suspicious because he is running around town with Billy Smith's camera in his back seat. So, suspicious guy. Sample number 2, murderer's DNA. So we have murderer's DNA from this homicide, and it was collected and was stored. And then we have the DNA from the victim, sample 3. And a lot of people say, well, why do you want to check the victim's DNA? This wasn't a suicide or anything like that. Well, what happens is, as you read about in the story, is that if somebody is murdered, they may fight back, in which case, in this example, the murderer's DNA, sample 2, is coming from underneath Billy Smith's fingernails. So he got some of his perpetrator's DNA, but we want to make sure that didn't cross-contaminate with Billy's DNA. So sometimes you also run the victim along with that, sort of as a control, okay? So we've got these 3 samples of DNA, and then we've got another vial. And if you can see in the camera there, this vial has the initials RE written on it. And RE, that stands for restriction enzyme. So this is a step where we are processing the DNA for a specific purpose. And that purpose is to allow unique segments of these individuals' DNA to become visible on a gel, okay? So what we're going to do is we're going to do the steps that you see in the lab, page 129. So page 129, they show us these 3 steps. They're talking about these 3 vials. They're talking about the vial of restriction enzyme. Tips, okay? And what I usually do, and I'm going to do this, is I open all the vials, including the restriction enzyme, okay? And use a clean pipette for each transfer. So you push down, you get the little pipette tip, and then we push down on the pipette, and you can see that there's some orange liquid in the pipette tip, perhaps. You can see that. That's the 10 microliters of the restriction enzyme. So that's what 10 microliters looks like. It's a very, very small, minute quantity of liquid. Okay, so I'm going to go ahead and put that into sample 3, push down a few times to mix, and then I'm going to close it, and then I'm going to discard this tip. Very important when we work with DNA samples is that we don't cross-contaminate DNA. So we use a fresh tip or a fresh tip for each transfer that we're going to do here. So now I'm going to take 10 microliters restriction enzyme into test tube 2, vial 2, mix that a little bit, close it, and one more. Now we have restriction enzyme into test tube 1. Okay. So we're allowing this DNA sample to interact with the restriction enzymes for the purpose of what we say digesting the piece of DNA. Okay, so first thing we're going to do is I'm going to try to make sure you can see what's happening here. Okay, so we talked about the agarose gel with the wells in it, and that we're going to be loading our DNA samples in that. So I'm going to very carefully remove these black stoppers, and then I'm going to place it into what we call the electrophoresis chamber. Let's see where is the best spot for you to see. I think like that, huh? Okay. Very carefully, I'm going to remove, do this close so I don't rip the thing here. Okay. So face this towards myself. I'm going to plug that little guy in there. There it goes. How's it looking? Can you see? Hopefully. Okay, so I put the gel in to the electrophoresis chamber, and now I'm going to do something. I'm going to actually pour a buffer solution on top, and the buffer solution is mostly water. There's some dissolved electrolytes in there, and the purpose of this is that remember there's going to be an electric current that flows, and the electricity flows better through water than it does through air. Okay, so we want this to be in water for us to see. Hopefully there's not too much of a glare on what you're seeing here, but I'll try to change the angle in a minute. Okay, samples. Now usually when we have you guys do this, we ask that you do some practice first, because it takes a steady hand. So future surgeons out there, this would be the technique to try to get a steady hand going. And I'll do a couple practice anyways because it calls for it in the drawing we're going to do. So I'm going to get a fresh pipette tip. Remember, we're going to use the micro pipettes. I'm going to use just some blue dye. And I'm going to fill my pipette with 20 microliters of practice dye. This is for the purpose of learning the technique. Okay, so what happens is you have to very carefully lower your pipette into the agar and add the dye to the well without getting much of it popped out there. And actually, I'm going to reuse my same tip because this is just practice dye, so I don't worry about contamination. And here, we're trying to get the DNA into the wells. But remember, this is just practice dye. So I'm doing 3 lanes of practice. Practice. I'm hoping you can see that pretty well. Don't want to move this around too much because I don't want my samples to pop out. But after we do the practice, we load samples 1, 2, and 3 into the gel. Okay, so I'm just putting them into the wells. So I'm going to start with sample 1, 20 microliters into the first well. Okay, and then I'm going to do sample 2. Into the second well. And then I'm going to do sample 3 into the third well. Change my tip every time, reduce contamination. So now hopefully you can see that I have all 6 samples loaded. Let's take you on a little journey to see what I'm seeing. How about that? Right, so you're able to see that I've got the DNA samples in there. 1, 2, 3, then practice is how that is loaded. Okay, next, we start electrophoresing. Yay! Here we go. We put a lid on it, and red and black leads, like your car battery, if you've ever seen your car battery. So we put the lid on it, and we have black. Black is the negatively charged electrode, we call it the anode, and then red is the positively charged electrode. We're calling that the cathode. Anyway, so the gel's going to migrate, the current is going to, move the DNA through the electrophoresis chamber from negative to positive charge. And that's going to happen because DNA is negatively charged. So I'm hooking up my leads. Let's back up a minute and see if you're able to see what's happening here. Oh, too much. I've got it plugged into an outlet right now. I'm going to hook it up to 125 volts of electricity. Zzz. And then we're going to watch. Hopefully you'll be able to see this. The magic. The bubbles. Here they go. We're in business. We're in business. I know. This is exciting. Look. Can you see something exciting happening? Let us take a little journey over here. Take a little journey with me, guys. Over here. Do you see bubbles? Do you see them? Can you see the bubbles on the chamber? Okay, so current is flowing. Current is flowing. And this takes about 15 minutes. It's been 15 minutes and our gel has been running. And it didn't run away. It's still here. Oh, joke. Okay, so we're going to take a look at the gel. And so I've turned off the electricity here so I don't electrocute myself. Wouldn't that be exciting, recording? Let's take off the current, okay, the lid. And then we're going to go ahead and take out the gel from the box there. So now the DNA has migrated, and I know you can't necessarily see, so let me go over here. And I'm going to go ahead and dump out the gel, okay, so that we can take a look at it. Let's make sure you can see what's important to observe here. Okay, so do you see that there are different colored bands that have migrated through our gel? And if you can see that closely, you should be able to tell that lane 1 and lane 2 have matching bands. Lane 3 does not match, okay, which was expected. Lane 3 was our victim, but lane 1 and lane 2 do match. And then we have our practice dyes as well. Okay, so let's wrap up this experiment by drawing what they're asking us to draw on page 133. So this corresponds to the gel that we just observed. And what we found was suspect had a blue band up here at the top that matched the murderer. Then we had a purple band about here. Then we had a pink band. I have red here, so red or pink. And then we had the yellow band somewhere around in here. And you may be wondering at this point, so when the DNA migrates through the gel, it changes colors? What's going on? Well, now I need to be real with you and tell you that none of that was actually real DNA. We don't actually allow the students to run this with real DNA because the chances of contamination are great. So rather, we provide you with these colored dyes as a simulation of real DNA. So that's why you actually see these different colors, as opposed to, in reality, they would all be either stained one particular color, or you do this over X-ray film, in which case they show up black and white. Oh, by the way, the victim had a yellow band here, and let's take a look here. They also had a pink band or red band there. And then the practice, might as well just put those in as well. These are not essential, but this was practice dye that you do in the experiment when you're learning the technique. So that's what your picture on page 133 should look like. And then that will help you answer the questions on page 134 of the lab manual. Now, I want to also show you page 126, the practice problem you're going to do there in the lab manual. Please remember to, of course, do all of the questions in the tan boxes. I didn't want to do all of them together in this video, so your instructors will be expecting you to do all of the boxes in the lab on those pages. So please do those. However, I get a lot of questions about this particular problem on page 126. So I wanted to look at that one with you. So here we're looking at a viral, something called a bacteriophage. And don't worry too much about the details there, but it's telling you that we are looking at the DNA of this virus, and we're looking at specifically fragments of DNA of different lengths. This is a little bit different than the experiment we've been doing in that they're using numbers that may not be familiar with for you, because previously we were talking about the GATA repeats, okay, the repeats at those short tandem repeats, those STRs that we find on chromosome 7 in particular. So now suddenly they're giving you huge numbers and you may not know what they're talking about. What they're showing you are lengths of DNA in a unit we call base pairs, or BP, because DNA is really long sequences composed of repeating units of DNA nucleotides, the alphabet, A, T, G, C, and so forth, right? So here, this is the full length of the lambda DNA, the name of this virus. Okay, this is their length. And what they're showing you is that there's this restriction enzyme, and this restriction enzyme is named HIND3, and it cuts here, cuts here, cuts here, cut. These are all cut sites. Remember, restriction enzymes are scissors. They're little molecular scissors that cut the DNA at unique locations. And they cut the DNA and produce all these fragments, okay? So what they want you to do in this problem is actually label the fragments in the gel diagram. So they give us this gel diagram and they tell us, it says indicate the length, the BP length of each cut DNA fragment shown on the left in the gel pictured below. Okay, so this is actually showing us the DNA separating. And of course, they want you to answer this question. In gel electrophoresis, how do DNA fragments of different size separate? They want you to understand these are different lengths. And the principle of gel electrophoresis is to separate the DNA by size. So the principle is larger DNA fragments migrate slower through the gel than shorter DNA fragments. Okay, so recall that's the principle we discussed earlier. So therefore, the bands at the top of the gel, because when it migrates, remember this is the negative charge and this is the positive charge. So DNA is being pulled in a downward direction, or diffusing rather, in a downward direction. And so what you want to do is you want to be able to label these fragments. Let me move the arrow this way. Okay, so the top fragment. This should be our largest DNA fragment of all these pieces. So you can imagine these all being cut into pieces. So what's the largest DNA fragment? That's right, it's the 23,130 base pair length sequence. So that must be this guy here, this band at the top. This must be 23,130 BP base pair. Okay, so the order, the way this works, the larger fragments migrate slower, and they do so in order, in length order. So therefore, the next band has to be what's the second largest. The largest is the 23,000 base pair length fragment. What's the next largest fragment? That's right, it's the 9,416 here. So 9,416. Okay, what's the next? 6,557 BP. Okay, next, 4,361 BP. Okay, we're getting down to the bottom here. Gotta move my plus out of the way. So what about these little guys? These are the smallest fragments. Remember, the larger fragments will be at the top and the smaller will be at the bottom. So next would be 2,322 BP, and then 2,027 BP. There you go. Okay, so you just need to label that in there. And yes, I just did it for you, but if you do a lab quiz on this, you may be asked to apply this principle to a practice problem or a quiz problem rather. Then part C, what is the approximate size of the DNA band in the far right lane? We're talking about this guy here. Well, it's a little bit higher up in the gel than this 23,000 base pair piece. So they want you to understand, basically, it would be greater than 23,130 BP. Really, it's the length of what would happen if you added up all of these fragments, and what would the size of this fragment be if we added it all up and we did not cut it with restriction enzymes. So add up all of those numbers, and that would give you the actual number of what that particular size would be. Okay? Hope that was helpful.